Thanks for downloading a 3CR podcast. 3CR is an independent community radio station based in Melbourne, Australia. We need your financial support to keep going. Go to www.3cr.org.au for more information and to donate online. Now stay tuned for your 3CR podcast.
Welcome to another episode of Lost in Science. You are listening to Lost in Science, another half hour of excellent science on your radio device. My name is Chris, and this week I will be talking to marine scientist Sophie Burgess about some research on scallops and what this means for sustainable fisheries of scallops. Sophie, thank you for coming into the studio.
Thanks for having me. I'm pretty excited to be here.
Brilliant. We will find out about you very shortly. But first I need to also mention that we also have a story from Stu, who will be speaking to Professor Tim Stenier from the Peter Doherty Institute for Infection and Immunity at the University of Melbourne. And he is talking about the Buruli ulcer, which is a flesh-eating bacterial infection that you may have seen popping up on the news. It's been infecting people around the Mornington Peninsula. Stu is talking to Professor Stenier about what that bacteria is and where it came from. Turns out that it's not as new as it might seem. Anyway, some disgusting bacterial stuff for you to look forward to there. Yeah, stay tuned. Yes, you are listening to Lost in Science, and I am speaking to marine scientist Sophie Burgess. Now, Sophie did some research at the Auckland University on the fisheries ecology of New Zealand scallops, and she is here today to tell us all about it. Sophie, welcome again to Lost in Science.
Thank you.
Now, I will be the first to admit that I don't know much about scallops, so can you tell me a bit, what do I need to know about scallops, what they are and what they do?
Well, they're really delicious. Yep, you eat them. They're a bivalve, which is a type of shellfish. And if you've ever seen SpongeBob SquarePants, there's these little birdie things that kind of flap around. They're scallops.
Okay.
Yeah, so if next time you watch SpongeBob and you see like the little birds, they're actually little scallop shells.
Are they like castanets kind of?
Yeah,
going through
so scallops can actually swim.
Okay.
And that's why they're birds, I guess, in SpongeBob.
So in bivalve, I assume that means it's got two shells?
Yeah, so it's got the two shells like clappers together. So, bivalves is two. They're usually found in the benthic environment, which is sort of the sandy bottom of deeper water. It's called the benthos. And they're really popular in New Zealand fisheries. Not so sure if they're as popular in Australia. And I've got some fun facts. They have eyes. They've got like 50 eyes. Next time you look at a live scallop, which is probably not very often if you're not a scientist like me that worked on them, they've got these really nice blue little eyes, and it almost looks like eyelashes as well, but it's actually their little filter-feeding tentacles. Okay,
so you're going to have to explain this a bit more for me, because I don't. So when you eat a scallop, or you get a scallop from the fish market, you have two bits, kind of. You have like the little round white bit, and then there's kind of a orangey squishy bit. Where is the rest of it? Like, where is the eyes and the tentacles and these kind of things?
That stuff gets removed when you eat it. So imagine a living scallop, not like the one that's on your plate. If you imagine the shell, the two valves are closed, and there's sort of like little squiggly ridges with little spaces between where I guess you could like imagine it like teeth touching each other, and then in between each of those gaps is a little eye. And then the tentacles kind of come out those little gaps, and that's what they use to suck in their food, which is algae in most cases. But what you see on your plate is basically the nice stuff. All the yucky tentacly stuff gets scraped out. And
all the organs and things.
The organs. But You do eat the gonads, which is...
what is that? How do I recognise that?
The orange part.
Okay.
Yeah. I know with
Is that the same in males and females?
So I'm not sure about the Australian species, but I know the New Zealand one, if you look at sort of the C-shaped orangey bit, sometimes it's half white and half orange, and that's like the female and male part. Can't remember which colour's which right now. And then the white, round, meaty bit, that's called the muscle. So that's what they use to keep their shell closed.
Right. So that the bit you eat is basically the muscle.
Muscle and the gonads.
Right. So
like the roe.
The roe. Okay.
Yeah.
Right. Okay.
Yeah.
Wow. And the rest of it, all these tentacles and eyes and things, they are discarded somewhere.
Yeah. So when you take them out of the shell, you've got to scrape out all the connecty stuff.
Okay. So you talked about these kind of eyelashy type of tentacles. So that's what it uses to feed?
Yeah. So
what does it eat?
They usually eat algae, which is just floating around in the sea, and little bacteria sometimes that are floating around in the water column. And basically what they do is they just wiggle their tentacles in the direction towards their... They don't really have a mouth. It kind of just goes into the stomachy part. Invertebrates don't really have all those complicated parts. And yeah, they just kind of wiggle their tentacles in and out, and it sucks the water and then all the bacteria and algae in there, and then they digest it and they're happy and they bury themselves and till they're next hungry, I suppose.
Okay.
Yeah.
So that's obviously you do know quite a bit about scallops. What was the focus of your research then?
The focus of my research was really to help us get a better understanding of the scallop fishery in New Zealand for population modelling. So that's how many scallops are out there in the population for us to fish. So my project was a fisheries-focused project. And I used a whole lot of data that was in a big database of the place that I did my thesis with, where they collected survey data of scallops for probably 20 to 25 years. And I basically went through that data and tried to make sense of it and help them make some predictions for future years. So that was kind of the most boring part, going through data. The fun part for me was actually going out on scallop boats. Now that I'm about to describe it, it doesn't sound that great. I got up at 3 a.m, hung out on a boat with like 2 guys that were really unshowered and stuff, because there's not really a shower on the boat and it's kind of tight living quarters. But that was fun anyway. You'd see like the sunset and, 3 a.m, not much is happening. So we went and did some scallop dredging, which is how you collect scallops by dropping it's kind of like a cage, I guess, with big teeth, and you drag it along the bottom of the benthos, which is the sandy bottom, and then you close it up, kind of like a bear trap, I suppose, and then all the scallops are inside there and the sand gets filtered out. So that was quite fun. But the most fun part was having my own baby scallops, which I did some research on in the lab.
And what did you do with those ones?
So what I was trying to do there is understand how scallops grow, so how fast they grow, and at what rate they grow, because that's something we didn't really know until my research. So I got some baby scallops. I measured them at the size that they were at the time that I got them. And then I had this really cool chemical called calcine, which is... It's actually a fluorescent chemical, so I dipped my scallops in that. They weren't harmed, they were fine. And then I just kind of let them grow, and I measured them at regular intervals, put them under the microscope, and I could see the fluorescent mark to measure how much they'd grown. Hopefully that makes sense. Kind of like tree rings, I suppose. They lay down individual growth rings. So that was the purpose of the scallops.
So like a new layer of shell every year.
Yeah. So with baby ones, the layers happen a lot more often. There's almost one a day. And that, because, when you're a baby, you grow a lot more than when you're old, usually. Mm-hmm. So that was what I did. But mainly it was feeding them, turning up in the morning, and they would kind of swim up spit water at me, and then I'm like, okay, I need to feed them.
So they recognise you? And
I don't think they recognise me, but they know because I
turned... It was feeding time?
Yeah, like I'd turn the filters off, and then they'd sort of come out from under the sandy stuff that was in the bottom of the tank. And then, because I would make up my own food, and I'd have to grow the algae in bags and sunlight and stuff like that. But when that went in the water, you could see that they could sense it with their tentacles, and then they'd sort of start swimming around. And did
they look like the birds on SpongeBob SquarePants?
Kind of did, yeah. They were really cute. Like, they actually look like that.
Okay, and so what did you find out? How long does it take a scallop to reach maturity then, for instance?
So to reach maturity, I know it's probably about 3 years. So by maturity, I sort of classify it as they're big enough for us to legally catch. So we know that takes about 3 years. But the research I was doing was only a couple of months, because obviously my thesis wasn't really long. So I just did maybe 3 months worth of growth tracking on the baby ones. And I sort of found that they put down a new growth ring once every 2 days. So that's kind of the growth rate that we gathered from my research. Yeah.
Okay. And so what are the implications then of what you found for the fisheries? Like, did you learn anything that can tell us about what the population is going to be like in the future?
Yeah, so it'll help us to make our modelling a little bit more accurate. So we've got all this data, and we're kind of guessing some of the things that we need to understand to estimate how many scallops there are. So we need to know how fast they grow, how many new babies are coming into the population, and how often they're dying and what's causing that. So we now actually have a little bit of an estimate of how fast they grow, because before that, in the models, we were just really putting an estimate from another bivalve species that I think Canada or something was using. We were just sort of copying their numbers because we hadn't had our own estimate. So now in the big complicated model, we can actually plug in they grow this fast, which is really cool. And hopefully the estimates are a bit more accurate. Whether or not that means we can catch more or less scallops, I don't know, because that kind of wasn't my job.
Did you see much population changes? We're looking through historical data. Was there?
Yeah, there was quite a change. There was a new scallop fishery found. Oh, it was probably in 2014. I did my research end of 2014, early 15, and there was a new fishery actually discovered, which is a really deep water scallop bed, which we hadn't really explored before. So the quota and the population numbers that we were allowed to take went up a lot once we discovered that unknown place. That was in the 2000s. And overall, the numbers, there's been a few sort of natural deaths in the population over time from disease and toxic algal blooms and things like that, and overfishing before it was sort of monitored properly. So it has changed from the data that I looked at. But yeah, that's due to lots of different factors.
Okay. Well, I suppose these are very important things. We do need to have some sustainable fishery as we rely more and more on seafood.
Yes, it is. I mean, things like scallops especially, they're one of the biggest exports out of New Zealand, as a lot of the other bivalve species are. So we, grow our economy by being able to fish them and export them. And we eat them as well in New Zealand. Okay. They don't all get exported. Mm.
Okay. Well, look, thank you very much for telling us all about scallops, their past, their present, and their future. And best of luck with your future research.
Thanks. Thanks for having me.
That was marine scientist Sophie Burgess.
Science, the final frontier. These are the voyages of Lost in Science, our ongoing mission to explain strange new words, to seek out new science and new explanations. to boldly go where no radio has gone before. Now, if someone told you that there was a flesh-eating bacteria out in the world, it might worry you a little bit, or it might make you think that was something out of science fiction. But in fact, there have been cases in Victoria of a particular bacteria which seems to have a very flesh-eating sounding effect on people. And I have got with me on the phone Professor Tim Stineer from the Peter Doherty Institute for Infection and Immunity at the University of Melbourne to talk about this bacteria. Thanks for joining us on Lost in Science, Tim.
You're very welcome, Stu.
Now, I did just mention a flesh-eating bacteria. This is basically what we're talking about, isn't it?
Yes, I think at its most sensational, that statement is actually true. This is a bacterium that gets under the skin, that grows in the fat tissue just beneath the skin, and does effectively eat your flesh. That's the alarmist part of it. I guess there's more to the story than that, but yes, it falls in that category of nasty disease-causing microbes.
So, what is this microbe that we're talking about? What's it called?
Well, its scientific name is Mycobacterium ulcerans. Now, the mycobacteria are actually quite there's more than 190 species in the genus Mycobacterium, and most of them don't cause any disease at all, but there are a few notable, notorious species within the genus, and one of those is Mycobacterium tuberculosis, causes human TB, and Mycobacterium leprae that causes human leprosy. So, there is potential in this genus for some very serious pathogens to emerge.
So it's got cousins that are undesirables as well.
That's right. That's exactly right.
Now, the name itself, Mycobacterium, myco comes from the word for fungus. Is there something about the bacterium that reminds us or is similar to the way fungi behave?
Not really. It's I guess that's a legacy of early days of microbiology. But no, it's a bacterium. It has the mycobacteria have a very thick cell wall that actually makes them quite resistant to sort of disinfectant killing, or even antibiotic treatments can be quite difficult with these bacteria. Yeah, so not really a strong connection with fungi as such.
Just an old-fashioned name then.
Mm-hmm, correct.
So this mycobacterium was relatively recently found in Victoria. When was the first case recognised?
Actually, relatively recent, I guess, but Australia has a reasonably long and proud history of discovery in this area. It was Australian researchers in the 1930s and 40s that first described Mycobacterium ulcerans as the cause of these nasty skin ulcers. And they did that by studying a small series of cases from patients that came from the Bairnsdale region in East Gippsland. And these were from people from farming communities that were presenting to local doctors, GPs in the area with unusual skin ulcers. And it was scientists at Melbourne at the Alfred Hospital that worked out actually that this was a new species of bacterium that the world hadn't seen before or recognised, and they called it Mycobacterium ulcerans.
Okay, so it hadn't been found anywhere else in the world, or is it present in other places?
Yeah, so actually what happened, so in the 1940s, Australian scientists gave a name to the bacterium causing these skin ulcers, but actually at the same time, doctors and researchers in Africa and Uganda and in the Congo, they realised that they were looking at skin ulcers caused by the same bacterium. They didn't call it Bairnsdale ulcer, they called it all sorts of other names, usually associated with whatever region it appeared in, but they're all caused by Mycobacterium ulcerans. And since then, actually, we know that the disease occurs in more than 30 countries around the world, but mostly these are tropical countries, and it's usually a disease of poverty. So the official name for the disease, I should say, is Buruli ulcer. This is the World Health Organization name for the disease. And as I said, 30 countries worldwide. Most of them are tropical, and mostly it's in sort of the rural regions of West and Central Africa. So this is a disease of the poorest of the poor, except around Melbourne.
So when did it start appearing around Melbourne? This is something that I've sort of popped up in the news.
That's right. So we've actually, there's a small team of us here in Melbourne, and colleagues down in Geelong have been studying this disease for the last 20 years. And actually we can trace back when the disease first appeared in Melbourne to around the mid-1980s. That's when we sort of first started seeing a few cases, and these popped up around Western Port Bay. And in the mid-1990s, we had quite a substantial outbreak at Phillip Island that then sort of flamed out after about 18 months. The disease is no longer found at Phillip Island. But since then, it sort of developed. It started appearing on the Bellarine Peninsula. And we've had sporadic cases, I should say, from around the Frankston area since the mid-1990s. But we had a big focus on the Bellarine Peninsula in Point Lonsdale, Barwon Heads, Ocean Grove. And then the bacteria and the disease have taken a trip across the heads and turned up on the Mornington Peninsula. And it's since about 2015 that the disease has really started to take hold on the Mornington Peninsula. And I don't know if you've seen in the papers, Stu, but we've had probably a doubling of cases since 2015, so that this year we're probably on track to finish the year with probably 200 cases, which doesn't sound like a lot, but for a disease that we were only seeing 10, 20 cases a year, that's quite a substantial increase, and that's where the alarm is.
Yeah, it's a tenfold increase in just the last couple of years. So where does it come from? How does the bacteria get from? How does it get into people for a start?
Yeah, this is a key question that's actually occupied researchers since the 1940s. What we call it the mode of transmission. How do people get the infection? And we don't have concrete data on what's going on here, but the research to date, all of the best evidence we have of what's happening locally, is that mosquitoes are playing a role in spreading the disease from contaminated environments. So, maybe gardens, the environment around where we're living becomes contaminated with the bacteria, and then mosquitoes are moving the bacteria from those contaminated environments to us when they bite. And you might say, well, how did the environments get contaminated? Well, we think that the Australian native possum, so the common ringtail possum and the common brushtail possum, are actually playing a role in amplifying the bacteria in the environment. We know that these two species shed the bacteria in their feces. They can actually get the infection themselves, and so they're playing a role as an amplifying reservoir for the bacteria in and around where we live. I might just add there, I don't want to vilify possums. They're a victim of this disease as much as humans are. But we do think that they're part of this transmission chain.
Okay, so it's sort of, can it be transmitted from person to person? Is it contagious in that way?
No, it's not. No, we've actually got reasonable evidence now from several epidemiological studies showing that there's no evidence of human-to-human transmission. This is environment to human. And our actually most recent experiments here at the Doherty Institute have shown you need very, very few bacteria sort of injected under the skin by a mosquito, and you can get the infection established. So, all the evidence to date points to no human-to-human, but probably environment-to-human, and we think that if you want to know how not to get a Buruli ulcer, what can you do to limit the chance of getting an infection, then the advice is protect yourself against insect bites, wash your skin if you receive any cuts or abrasions while gardening perhaps. Make sure you wash those and treat them with disinfectant promptly. Cover up when you're outside, sort of common sense things you would do anyway. I guess that, for the people listening to this program, Stu, I'd like to circle back to your introduction when you talked about flesh eating. It sounds really alarming, but the point to remember, this is still a rare disease, and it doesn't kill you. We have good antibiotics to treat it, and we need to just make sure people get diagnosed quickly. And there are some simple precautions like avoiding mosquito bites and washing after gardening that should reduce the risk of infection.
I guess, as you say, they are pretty common sense things. Nobody really wants to get bitten by mosquitoes or get any kind of infection from cuts and abrasions. So it's sort of common sense stuff there. If people suspect that they might have contracted it, what would you suggest that they do then?
Yeah, so I think you just need to suggest the diagnosis to your GP.
Yep.
Say, look, could this be Buruli ulcer, Mycobacterium ulcerans infection? And there's a very good diagnostic test, again developed by the team of researchers here at Melbourne, that can very quickly and with a high degree of accuracy tell you whether this is a Mycobacterium ulcerans infection. So good diagnostics, and once that diagnosis is made, then there's a well-established treatment using antibiotics. Now the antibiotics are quite potent, but they are effective. And so once you get the right diagnosis, then you're on the path to recovery.
So it sounds like maybe the media is inflating the story a little bit by calling it a flesh-eating bacteria, but it's still, it's a worry for people who maybe are holidaying at the seaside, which will probably be increasing in the coming months as we move into summer.
Look, I think it's a rare disease. It is increasing, but for the individuals that develop this infection, it's a serious infection. Like, it's not. I guess I don't want to trivialise it either. So the chances of any one of your listeners developing this disease is small. But if they were that unlucky, then it can be quite a serious infection. So it's one of those, weighing up the risks. The risk is small, but if you're unfortunate enough to pick up this infection, then it can be quite nasty.
Okay, well, I guess the statistics say that you're probably not likely to get it, but it is worth taking precautions. And as I said before, nobody really wants to get bitten by mosquitoes, this not being the only thing you can contract from mosquito bites.
That's right. That's right. It's good common sense anti-infection control practices. That's right.
Yeah, so if we just keep an eye on mosquitoes, try and control them in our local area, and I guess wear the insect repellents and loose clothing so that they can't bite people, that's probably the best way to avoid it.
Definitely. That's right. No, fully support that advice. And if you go to the Department of Health and Human Services website, they offer similar advice.
Well, fantastic. It does sound slightly scary, but it sounds also manageable. So thanks for joining us on Lost in Science, Professor Stenier.
You're very welcome, Stu.
And that's it for another episode of Lost in Science. Lost in Science, it is recorded at the studios of 3CR in Melbourne, and it airs across Australia on the Community Radio Network. We would love you to get in touch with us. If you want to do that, you could email us at lostinsci, that's SCI, @gmail.com, or you can find us on Facebook, where you can friend our page. We are Lost in Science on 3CR, or you can look us up on Twitter. We are Lost in Science 1, I believe. Or you can look up our podcast. If you are listening to our podcast through iTunes, then please take the time to go over there and give us a good rating and review, because that will help other people find this wonderful science programming. Or if you listen to us on the radio, you can find us again same time next week. And once again, Stu, Manisha, Claire and Chris will get
Lost in Science!
Thanks for listening to a 3CR podcast. 3CR is an independent community radio station based in Melbourne, Australia. We need your financial support to keep going. Go to www.3cr.org.au for more information and to donate online.